All right. Good morning, everyone, and thank you for joining today's webinar entitled CRISPR-Edited Mammalian Cell Lines, Advantages, Applications, and Tips for Using this Revolutionary Technology. My name is Laura, and I am the Marketing Specialist for the CRISPR services here at Genscript. I am pleased to introduce our presenters today. We have Dr. Wensheng Jia and Dr. Sean Zhu, who are senior scientists here at Genscript with years of CRISPR experience. So the CRISPR-Cas9 technology has quickly become one of the most exciting gene editing techniques available and has revolutionized the generation of recombinant cell lines. So in this webinar, we're going to describe the advantages of CRISPR over other technologies, and we will also be going over some strategies for optimizing CRISPR for your research. And then at the end, I'm going to discuss some of the CRISPR cell line services that Genscript can provide to accelerate your projects. So if you have any questions during the presentation, you can submit them by typing them in the questions field that you see on your screen. And at the end, we will do our best to answer them after the webinar. Frequently asked questions along with their answers, as well as the webinar itself, will be posted on our website at www.genscript.com. And finally, at the end of the presentation, you'll receive a brief survey. We encourage you to fill out the survey as it'll help us better design and optimize our services to meet your needs in the future. And so now with that said, I would love to turn the presentation over to Wensheng.
Hi, to start, I want to briefly overview what I will be covering during today's webinar. First, we'll be discussing some of the basics of CRISPR-Cas9 editing technology, including how it originated and the important components of the system. Next, we will describe specific advantages and applications of the system. Finally, we will spend time discussing some helpful tips for generating CRISPR-edited cell lines, and at the end, we will conclude with the question and answer section. In the first part, we will briefly describe the basics of CRISPR-Cas9 technology. So, to begin, what exactly is CRISPR? The CRISPR system was first identified as part of the adaptive immune system in bacteria. In the late 80s, researchers observed curious repeat and non-repeat sequences in the bacterial genome. These repeat sequences were later cloned as cluster regularly interspaced short palindromic repeats, or short sequences of viral and bacteriophage DNA that becomes cleaved and inserted into the bacterial chromosome after infection. When the bacteria is infected again later after the same virus, the CRISPR complex is translated and cleaves the DNA. For editing mammalian cells, three components are essential. First is the Cas9 endonuclease. The Cas9 endonuclease generates double-strand breaks into the chromosome. Ending nuclear localization signals improve its function in eukaryotes. The guide RNA, which consists of both CRISPR RNA and tracer RNA, combine together to form a chimeric guide RNA with the same function, which is to guide Cas9 to targeted gene sequences. In cases where genes are being knocked in, a donor DNA sequence will also be needed. Following transfection, the Cas9 gRNA complex will bind to the target sequence and then make double-strand break, followed by NHEJ, which is homologous and joining or donor DNA template-induced HDR in knock-in cases. Following transfection, the Cas9 gRNA complex will bind to the target sequences and then make double-strand break, followed by NHEJ or donor DNA, as mentioned. After double-strand breaks are introduced by Cas9, there are two ways they can be repaired. In the absence of donor DNA, double-strand breaks will be repaired by NHEJ, creating an indel or gene knockout. In instances where you want to introduce a specific change within the genome, for example, upon mutation that causes a special amino acid substitution in the protein product, a donor template will be needed. The donor template, which can be delivered either in plasmid or as single-strand oligos, will be used for HDR after Cas9 creates a double-strand break. Now that we've covered the principles of CRISPR-Cas9 technology, next I will discuss the advantages of CRISPR-Cas9 technology compared to other genomic engineering systems. Until now, three main genomic engineering systems have been identified. The first system we will discuss is zinc finger nuclease. ZFN is consistent of zinc finger DNA binding motifs in beta-beta-alpha configuration, which recognize 3 BP segments in the DNA. The system also includes a nuclease, which targets the cut these segments. Another common editing technology includes transcription activator-like effector nuclease, TALENT. In this system, proteins are designed to contain DNA binding domains that recognize specific DNA sequences down to the base pair. This nuclease component will also target and cleave these DNA sequences as they do for ZFN. Finally, the CRISPR-Cas system exploits a 20-nucleotide CRRNA fused to a tracer RNA and the Cas9 in the nucleus that recognize specific sequences down to the base pair. Compared to the ZFN and the TALENT technologies, CRISPR has many advantages. Firstly, the CRISPR system has the broader use by any lab, as less molecular biology expertise is required. Both the ZFNs and TALENTs require the design of proteins that can recognize the short DNA targets, which is very labor-intensive and expensive. In addition, the efficiency of this system is quite low, requiring potentially multiple rounds of editing to achieve the desired knock-in or knockout. CRISPR, on the other hand, only requires design of the guide RNA Cas9 complex, which is quicker and easier than engineering proteins, and with the ability to target multiple genes simultaneously, is much more efficient. Overall, the CRISPR-Cas technology has made gene editing possible for any lab at any budget and expertise. Now we are going on the third part, the application of CRISPR-Cas9 technology. One common application for CRISPR-Cas9 in cell lines is to knock out a protein coding gene. If we want to knock out protein coding genes, both gRNA and Cas9 vectors are needed. One Cas9 is required to target the protein coding sequence, and the Cas9 is guided to this target. The gRNA and the Cas9 complex will generate a double-strand break, and in the absence of donor DNA, the double-strand break is required by NHEJ to create an indel. The knockout clone is then isolated, and from this clone, you can confirm whether a form-shift mutation has destroyed the protein coding frame. Successful knockout of the target gene is ultimately validated by the Sanger sequencing and Western blot. If you are trying to knock out non-coding DNA, the process is a little different. In this case, 2 gRNAs are needed to cut the flanking sequences of the non-coding DNA. Similar to the method presented on the previous slide, After isolating the clone, you can confirm knockout by fungal sequencing. This method can be applied to knockout of a promoter, microRNA, or transcription factor binding DNA. If you'd like to introduce a specific change within the genome, for example, a site-specific point mutation, it is necessary to supply a donor template that can be used for HDR after Cas9 creates a double-strand break. HDR templates are delivered as single-strand oligos. When designing this ssODN donor, a minimal homology region of 40 base pair and preferable 90 base pair is recommended on either side of the modification. Another point to pay attention to is avoiding complementarity between the gRNA and ssODN. HDR efficiency can also be increased by inducing a silence mutation on the ssODN or by choosing a guardian that is not complementary to the ssODN. Another application for CRISPR in mammalian cells is to integrate targeted genes at specific sites on the chromosome. In this case, the donor template for HDR may be delivered in plasmid. Typically, the plasmid donor should have two homologous arms on each side around 0.5 to 1 kb, blocking the desired insertion or mutation. To assist in detecting successful HDR and codifying knock-in efficiency, donor templates are often designed to include several synonymous mutations. To prevent the cleavage of donor templates or of the genomic DNA after successful HDR, the donor template should be designed with mutations in the PAM sequence. In the next section, we will describe some useful tips when you are planning your CRISPR-Cas9 experiments. In designing your CRISPR experiments, there are 5 methods to improve the reliability of your project. These include choosing the right gRNA and Cas9 construct, choosing the best delivery method, evaluating the gRNA sequence to ensure accuracy, enriching the transfected or transduced cells, and finally, optimize HDR efficiency. When choosing a gRNA Cas9 construct, you can use Cas9 or a pair of Cas9 nakeds. Cas9 nakeds have mutations in the catalytic domain of Cas9, causing them to create single-strand breaks instead of double-strand breaks. Targeting this Cas9 to two loci within close proximity causes Cas9 effectively nick rather than cleave DNA to use single-strand breaks, thus effectively increasing the specificity of target recognition. Proper promoter selection is dependent on your cell line, since you want to ensure the plasmid and the promoter suit to work in the host organism. Commonly used promoters in mammalian cells are CBH, EF1R, CMV, CAG, and SV40. The promoter activity differs between cell lines. Therefore, you want to choose the promoter with the highest activity in your cell type. Another factor in gRNA Cas9 design is the selection method. CRISPR plasmids typically contain selection markers such as gene-conferring fluorescing protein for easy visualization by FACS or antibiotic resistance to enrich the positive selection. Both GFP, GFP, or RFP expression and antibiotic selection can be used to identify positive population or clones. Once you have designed the construct, then you need to choose the best gRNA and Cas9 delivery method. For easy-to-transfect cells, lipovaccine or nucleovaccine can be used for delivery of Cas9, gRNA, and donor DNA. This method is simple and is very efficient for easy-to-transfect cell lines and larger insertion vectors for knock-ins. However, it is not suitable for hard-to-transfect cells. Well, for hard-to-transfect cell line or primary cells, lentivirus or adeno-associated virus-based delivery of plasmid is preferred. It will provide higher delivery efficiency for prime and not-binding cells as well as common hosts. But there are limitations in viral packaging, including the inability to deliver larger insertion sequences or long donor genes. Well, before starting your experiment, it is also suggested to evaluate the gRNA sequences so that the most optional gRNAs are being used. Commonly, we evaluate gRNAs by examining the cell pool transfected with gRNA Cas9 plasmid. There are two main methods for evaluation: the nucleotide mismatching assay by sequencing and the surveyor assay. The nucleotide mismatching assay by Sanger sequencing is a simple technique and may give a false negative when the efficiency is too low to detect by sequencing. This assay typically works best for cells that are easy to transfect. Alternatively, the surveyor assay can easily identify mutation or indels, and digestion with the mismatch specific DNA and the nucleus work well for all cell types. Enrichment of transfected or transduced cells by antibiotic selection or FACS sorting can increase ratio of positive genome-edited clones in the cell pool, especially for the hard-to-transfect cell lines. FACS sorting is more effective for separating the populations and can also eliminate dead cells. But performing FACS sorting may limit your experiment because of the need for special instruments. It is a low-throughput approach, and the sorting process may be detrimental to some cell types. Alternatively, when antibiotic selection is chosen, no instrument is required, and high-throughput selection is allowed. A disadvantage to this method is that gRNA and Cas9 may be integrated into the host genome. If antibiotic selection is selected, pyrimycin is an ideal choice because selection may be finished in 3 days, avoiding stable integration. Improving the specificity of CRISPR genome editing is important to avoid undesired mutations. In regard to the gRNA itself, although 20 base pair regions were initially used, it was observed that mismatches were tolerated most often in the 3' end of the gRNA. Researchers in the joint lab found that gRNA with 17 or 18 nucleotides of complementarity function as efficiently as 20 base pair sequences to induce mutations, and this showed reduced mutation effects at close match off-target sites. The strategy for improved gRNA and Cas9 targeting specificity is to exploit a pair of guides that targets very nearby regions. The mutation in the catalytic domain of Cas9 causes it to create single-strand breaks. Targeting this nickase mutant to loci within close proximity but occurring on opposite strands of the genomic DNA causes Cas9 nickase to effectively nick, thus ultimately increase DNA specificity and improve target recognition. Whether a mutation is off-target or not could be tested using different clones generated with the different gRNAs. If same mutations are detected, it is surely on-target mutation. And finally, optimizing HDR can also improve the overall efficiency of the CRISPR system. HDR occurs much less frequently than NHEJ, and therefore inhibiting of the NHEJ pathway can drive double-strand break repair towards the HDR pathway. Today, there are three techniques that have been used to suppress the NHEJ pathway. One method is to inhibit protein expression by shRNA silencing. By inhibiting the expression of key proteins that provide the green light for NHEJ, you can improve HDR exponentially. Enzymes such as DNA linkage 4 are also active during the NHEJ process, and consequently, inhibitors have been observed to increase HDR-mediated genome, adding up to 19-fold in the mammalian cell life. Similarly, co-expression of AG4 proteins has also been observed to favor HDR. Finally, HDR can be further improved if the PCR fragments for donor DNA have combined homology region more than 1 kilobases. For more information on how HDR efficiency can be optimized, the recent Nature Biotechnology publication list here can provide some additional details. So finally, our marketing specialist, Laura, will take a few minutes to summarize our services here at Genscript.
Great, thank you so much for your presentation, Wenshang. So, as she mentioned, I just wanted to take a few minutes to describe some of the many CRISPR mammalian cell line services that we offer here at Genscript. So, currently, we offer both knockout and knock-in genome editing services that can target any gene in any mammalian cell line. And so those are just, you can see in the first two rows here. So these services will either use non-viral or viral transfection methods to optimize CRISPR delivery into cells. And the turnaround time for these services varies depending on your project, but they'll start at about 16 weeks and $8,000 per cell line. And at the end of this time, we will deliver a single clone with the target sequence validated, as well as a detailed report of the gRNA design and sequence. And then I also wanted to take a moment to share with you some services that we have that are coming soon in the next month or two. So one we have is called a guaranteed knockout cell line service. And this is going to offer lower pricing for easy-to-handle cell lines. And we will also provide a knockout stable cell pool service with lower prices and turnaround times as well. So if you'd like some more information on these services, or if you want to find out when these new services will be offered, then please visit our website at www.genscript.com at CRISPR.html. So in addition to our editing services, we also offer extensive preliminary and post-editing analysis if required. So as part of the preliminary editing service, we offer confirmation of sequences prior to gRNA design. We will perform investigation of gene copy number into the host cells, as well as an off-target analysis by Sanger sequencing. And then for our post-editing service portfolio, we can offer RT-PCR and Western blot analysis to confirm knock-in and knockout at the gene and protein level, respectively. And then we can also offer additional assays, including growth curve characterization as well as functional analysis as needed. And so in summary, Genscript's CRISPR services are unique in that we can offer one-stop services, everything from assay design to functional assays. As a market leader, Genscript was one of the first companies to offer competitively priced, customized CRISPR service, and more recently has become one of the few companies to offer both viral and non-viral transfection techniques. We have extensive experience with a variety of immortalized and tumor cell lines with a record of high efficiency regardless of the cell type. And with a license from the Broad Institute, we have been able to remain a leader in CRISPR-based genome editing. So in addition to our cell line services, we also offer a variety of other CRISPR services, and I have them listed here. These will include CRISPR plasmids. We also offer GECKO and SAM gRNA libraries. And then we also have just introduced a service, CRISPR service for microbial genome editing if you're interested in E. coli recombinant strains. And so if you'd like more information about these services, please feel free to visit our website that's listed here, www.genscript.com, CRISPR.html. So before we start taking questions, I just wanted to tell you about some of our upcoming webinars. On October 22nd, we'll be hosting a webinar on efficient stable cell pools and cell line development for large-scale antibody and protein production for those who want to know a bit more about our protein services. On October 29th, we'll be presenting the webinar examining the components of your peptide sample with AccuPep QC for anyone who routinely works with custom peptides. And then also upcoming is on November 5th, we'll be presenting a webinar entitled Large-Scale Genome Editing for Metabolic Engineering on E. coli, which introduces our newest CRISPR service in microbes. So please feel free to register for any of these webinars for free by visiting the website listed here. Great, so this concludes our webinar on CRISPR gene editing and mammalian cell lines. So if you have any questions, please feel free to ask. We'd be happy to take them. Again, to ask a question, simply write it down in the question field that you'll see on the right side of your screen. All right. Okay, so thank you again. So what I'll do is thank you for your questions. At this point, I'm going to turn it back over to our scientists who can help you guys out. So, Wenshuang, I'll turn it over to you.
Okay.
Hello, this is Sean. So nice to have a talk with you in this webinar. So right now I got one question. So the question is, can one screen engineer the cell clone for homozygous deletion efficiently with the Sanger sequencing? Is this state of the art? So for this question, so I think the Sanger sequencing is the best way to screen the cell clones for homozygous deletion. As you know, so we will generate a lot of cell clones, generally more than 100 clones, and then we will copy the clones from the 96 well plates to 24 well plates, and then we can amplify the target fragment using the PCR and then do sequencing. So I think it's the simple way, and we are using this method to do the screening in high throughput. So this is the first question. I hope my answer, my reply to your question. So what's the second one? So the second question I got CRISPR was described here to edit mammalian cell lines. Can this system also be used to edit the genes of virus to a swamp of a wild-type gene with an alternative gene? Okay, for this question, I think currently I have never heard the CRISPR technology could be used for virus genome editing. As you know, the CRISPR Cas9 is adaptive system to destroy virus. If you want to edit the genome of virus, I think you need to use a method to use a system to repair the virus genome. As you know, in the mammalian cells, when the CRISPR-Cas9 complex cuts the genome, and then the NHEJ pathway or the HDR pathway will be induced to repair the genome. So for the virus, so if you don't have a system to repair the genome, so it will be very difficult. Let me try to get the next one. Okay. The next one question: does nucleotide mismatching assay need to do after the plasmid was constructed or before? My answer is no. So this nucleotide mismatching assay is for cell pool examination. So as you know, the nucleotide mismatching indicates the indel mutation has been generated in the cell pool. So for the plasmid, I think what you can do is to test the DNA cutting efficiency in vitro, but it is impossible to detect nucleotide mismatching assay with just plasmid. Okay, so this is my answer. So what's the next question? So the next question is how do you establish the gene copy number in the host cell? So for this question, my answer is to use the qPCR. As you know, the qPCR can indicate the gene number in the genome. So if we use a standard cell line which you know how many copies of the gene in this cell line, and then you can evaluate the gene copy using the qPCR. So this is the simple way, I think. So what's the next? So the next question is can transfection integrate into genome? This is a very good question. So actually, I think it will be very difficult to avoid integration of the plasmid to the genome. so it will be difficult to control, but you can use some selection marker to avoid those clones, such as using a GFP expressing plasmid. If the GFP has been integrated in the genome, you can observe the clone under the microscopy. If you observe the GFP, that means the plasmid has been integrated in the genome. So I think this is what you can do, especially for those cells with very high transfection efficiency. The integration efficiency will be very high. Okay, this is my answer. So what's the next question? Can you use CRISPR to insert an in-frame GFP tag to study localization of a specific protein? So my answer is absolutely yes. So actually we can use, so we need to do a knock-in in the cell line. So we can, using a donor plasmid which contains the GFP tag. So if you're using a gRNA specifically cut your target gene, and then the donor plasmid contains the homology arm, and with the GFP tagged at the end of this gene, and then you can get the clone with the GFP tagged clone. Okay, this is my answer. Yes, you can do it. So what's the next? Okay. Is there any more question?
Great, thanks so much, Sean. It looks like that is the end of the questions that we've received. Thank you guys again for attending this webinar, and I just wanted to reiterate, if you do have any more questions, you still have a chance to ask them. You can either email our scientists at the emails listed here, or please feel free to visit our website. So I have here the website that refers to our technology resources, which will have a lot of answers to frequently asked questions. And it'll also have some other resources available to you as well, including we also have a CRISPR handbook on this website that can also give you some good insight into CRISPR and how you can use it in your research. So with that, I would just like to thank our presenters again. Thank you very much for your time, and thank you as well for attending our webinar.
Thank you.